adept in sort of sequestering ephemeral sources of pollen. And because of this great power they have, they often sort of exploit resources that native bees would also like to use, and by that exploitation may or may not prevent native bees from using the resource, okay? So we're kind of looking at competition, and competition can take place in two different ways. The exploitation I was just kind of talking about, whereby somebody uses the resource and then somebody else can't use it, or interference, where there's some kind of a direct interaction that alters another bee's ability to use that resource. So if I wanted to anthropomorphize it, maybe like I've got my can of SpaghettiOs and I set it down in the kitchen and walk away, and later on Abe comes by and picks it up and takes it from me, and then I have to go look somewhere else further away for some SpaghettiOs. Or maybe I go into the kitchen and go after my SpaghettiOs, and then Abe hog-ties me and takes them. That would be, the latter is your interference, all right? Oops. Right should not advance.
No, right isn't advance. Well,
oh, there's two rights.
Don't make her wrong. They do make her wrong.
Okay, so this is an example of one of our transects, and these little stakes you see in the ground, a little cup inside of them, are our bee bowl setups, which I'll talk to you about in just a second. So the transects themselves, they're 30 meters in length, and the bee bowls themselves are spaced 5 meters apart. So there's 7 of them to a transect. And we're going to be looking for a distance effect. So after we do some sampling and see what kind of biodiversity is out there, we're going to bring in a beehive and see how that alters the competition in the area. So the bee bowls themselves, what they are, is a rebar stake with a PVC tube on top of it, and there's a little coupler and a dish in the top. And then into that dish we set another dish that's painted with a fluorescent color, blue, yellow, or white, that attract the bees. And we fill that with a splash of soapy water and leave them in place for about 12 hours. And the bees come by, they're attracted to the color, they see some liquid in there, they go down for a drink, slip in, and then drown. And later on, we come by and collect them and then analyze them later in the lab to see what's out there.
Yeah, and we're also trying to get an idea of just what's flowering out there. So weekly we're going to be conducting basically just plant surveys of what generally is out there. Trying to get an idea, basically the main purpose of it would be to see like, okay, are there more native bees out and active while this plant's flowering versus this plant? Try to see if there's any correlation. And we're quantifying these on a 3-point scale, rare, common, and dominant. Like rare being there's a few here and there, common is here and there, and dominant is like fused patches. So that's what we're doing. And then we also, along with our exploitative thing, we also set up this high stakes resource, which is basically just an array of these nectar cups that are going to have a dental wick inside them and a honey solution. So then the bees will hopefully come to that solution, feed, and we're going to try to observe those for about an hour or so and see, like pinpoint the visitor, see how long he stays there, and then if he comes back or not. And also, try to monitor interactions at the feeder, so like B2B interactions, being that maybe a native bee's feeding, honeybee comes in, pushes them out, and then the native bee goes away.
We're going to start off by kind of training them to the area where we are going to set the array. So like right here, Abe's got one of the arrays that we're going to use. And over here, we're in the exact same spot, but we've got this little trainer out here, which is just a big dish full of honey solution, 25% sucrose or so. Then we set a big old sponge inside of it that's soaked in the solution, and it's just a whole bunch of honey that the bees can come by. They're like, okay, I know the honey's here, and then later on when we're actually doing our observations, we'll be using this array. Okay, so the things that we have still to come, so I talked earlier about bringing in a hive, and I talked about our transect. So the hive, we're going to set it right in the center of the transect, which is 700 meters in length. And then what I'm going to be specifically looking for is essentially a distance effect. So from that hive, the further we get away, we would expect that if honeybees and native bees are exploitative, well, if there's exploitative competition occurring, that the further we get away from the hive that we've introduced, the more native bees we should expect to collect. And that's the hypothesis that I'm testing. Abe, on the other hand, there's not a whole lot in the primary literature about interference competition between honeybees and native bees. So that's kind of up in the air still as far as what we're going to see, and it'll be fun. And we haven't gotten a whole lot of sampling done yet, but our infrastructure is up, and we expect good things to come.
Any questions?
Yes.
Have you thought about like videotaping if you do find that there's
We've
interaction?
thought about that. We figured that it'd be better just to sit there and, because we don't really know what we're going to see at this point in time, so we're kind of playing it by ear and kind of seeing what's there, but that's also a good idea. We've thought about it. We just haven't necessarily decided.
Yeah. Anytime you have to parse through film data, it can be.
Well, I wasn't necessarily thinking of filming the whole thing, but if you do find that it's something new,
Yeah, definitely.
make a nice YouTube video.
Yeah.
For sure.
Lindsay?
A question about the beagles. How are you ensuring that you're not having all sorts of unnecessary deaths of other species?
There's really no way to ensure that.
I mean,
Is there a way that you could put a lid with an opening of appropriate size to prevent like butterflies and larger pretty things?
well, the way I think about it is, we're gonna catch some other stuff. We catch a lot of flies, a lot of butterflies, and the number of deaths is really negligible. I mean, we're putting such a tiny dent into what's out there that we're not overly concerned.
Well,
But
I know that you're right. I don't think you're going to kill anything before you destroy the population, but, for the tender heart over here, is there a way that you can make it go smaller or something? I don't know, I'm just
That's
curious.
so-
yeah, that's definitely an interesting idea. I don't want to. These have such various sizes, so I don't want to deter a bumblebee from coming by in order to get a little sweat bee or something like that to enter the bee bowl. So something that we can consider, but we haven't really done so yet. Jeff?
Is there a reason for using different colored flowers?
There's a lot of variation in literature about which one is like the best, but it's literally fluorescent blue, yellow, and white. So we just figured we'd use all of them to cover the whole spectrum.
Okay. And you see there's no blue here. Couldn't get any of the.
Yeah, they didn't have to paint that day, so
Yeah.
yellow and white it is.
Yes.
Are you going to record the color of the dish and the number of individuals that you find in each? Maybe then you can say which one is the best?
Well, there's been some scientists out there who have tried to do it. Some much more qualified than us even, who have found that it's really overwhelming to attempt to see which color is best, and it's kind of negligible. The
For what we're
differences. So,
specifically trying to look at.
yeah. So really the kind of the the current standing in the literature is that they're all equally effective, all three colors. Questions?